Hey guys, we're going to go through the notes about cellular transport. Okay, so this is our idea of entropy. So this is a measure of disorder in a system. So you can think about if you have your yard at your house, right? You have to constantly put energy in it because over time it's going to naturally go towards disorder. This is the same idea that we have in all living systems, is that if there's no energy put into it, it's going to go into disorder. Okay, so because those atoms are going to spread apart and unless that energy again is added to the system. Okay, so you can think about a desk even. It's going to, if you don't put energy into organizing your desk, it's just going to be chaotic and disordered. Same here. It naturally goes and spreads out because of that disorder. This is our idea of entropy. Okay, and this is why we have something called passive transport. So passive transport, it's when our substances are going to diffuse, we call it with their concentration gradient or down their concentration gradient, meaning it's going from high to low concentration. So it's able to do so naturally because it's following that natural entropy. And it does not require any metabolic energy, so no ATP is required. Going from a high concentration to a low concentration, just like rolling a ball down a hill. It's very easy to do. Okay, we have two types of passive transport. We have simple diffusion, or sometimes just referred to as diffusion, and that's when our small molecules, these are again going to be our uncharged, our non-polar molecules, can go straight through the membrane through those phospholipids. That's diffusion or simple diffusion. Facilitated diffusion, this is what we refer to it as when we have those larger molecules or charged molecules that can't pass through and they need that help by going through a membrane protein. So we use facilitated diffusion. It's still diffusing high concentration to low concentration, but it is being helped or facilitated by these channels or proteins. Okay, so our diffusion again is that movement down a concentration gradient. Will go from high concentration to low concentration. And certain molecules can diffuse through the cell membrane, and some others need help with assistance. So which kinds of molecules can diffuse? These are again our small nonpolar molecules that can diffuse straight through the membrane because of that also nonpolar region of our membrane. There we go. Okay, so our diffusion here, we have a rate of diffusion. It is dependent on 5 factors. Size of the molecules, okay, so smaller molecules will diffuse faster than bigger molecules, okay, or vice versa. Larger will diffuse slower. Temperature also plays a risk or a factor, right? Increasing temperature is going to increase the speed of our molecules moving. So if we have a higher temperature, we're going to have a higher rate of movement. The steepness of the concentration gradient. So if we have a really, really concentrated area and a really low concentration area, it's going to move a lot faster than if we have two regions that are more similar to each other. And then the last one is the charge of the molecules involved. Oh, 5, sorry, there were 5, and the pressure as well. We increase the pressure, it's going to push those molecules to diffuse faster. Okay, so now we're going to get an active transport. This is the opposite of our passive, and here we're going from low concentration to high concentration, so it's the reverse. So here you can see we have molecules being pushed into one area. Even though it's highly concentrated, it continues to push more in that area. This does require ATP because it's not following our entropy, so it's going to require additional ATP to have this happen. Okay, so this is like going up a hill, low concentration to high concentration. Pushing a ball up is going to be a lot harder. It's not happening naturally. Okay, so our active transport, we have some examples. One is a protein pump. Similar idea to our protein channels, but this is made out of protein, but instead of just having a channel or a pathway for our molecules to go through, here is actually going to be actively pumping. So this is happening by phosphorylation, which is adding a phosphate from ATP onto that protein shape, and it's going to activate our protein pump. So here we have an unphosphorylated protein, a protein with no phosphate, and then we're going to add phosphate from the ATP and make a phosphorylated protein. Okay, that's how these pumps like this sodium-potassium pump works. We use ATP, and then that energy from it changes the shape of our protein so it's able to pump in the correct way. Okay, so we have a couple examples of these active transport. One is a proton pump. Okay, so this is helpful in our cells. So the idea is that it's pumping protons, right? So we have our hydrogen ions, and they're getting pumped from a low concentration to a high concentration. So it's getting pushed against its concentration gradient to make a really highly concentrated area. Okay, we can also get the idea of co-transport, co meaning two. So here we have two molecules being moved as opposed to one with our proton pump. Okay, we can have symport, sym meaning the ions are going in the same direction, and antiport meaning the ions are going in opposite directions. Okay, so here's an example. Uniport was like that hydrogen pump we just had. Symport, two ions are going the same direction, and an antiport, one will go one way, one will go the other. Okay, so our example of a coport pump is our sodium-potassium pump. This is making a concentration gradient, so a high concentration of sodium outside and a high concentration of potassium inside. Okay, so this is showing here where we have our sodium binding to a specific active site. It needs to be phosphorylated to activate and use energy to have this protein function, and then it will have a conformational change in the shape and push the sodium out from one end. Same idea will happen. Sodium, once the sodium is released, the potassium can come in and bind, and then it can be transported to the other side. Okay, so here's showing kind of in a movie type what's happening, where it is phosphorylated from that ATP, and then it is able to pump it to the correct side. So we end up having one side that has lots and lots of sodium and one side that has lots and lots of potassium. And we will go into these examples in photosynthesis and cellular respiration next unit. So these will come into play again, our sodium potassium pump and our hydrogen pump. Okay, this also gets at our idea of membrane potential. Okay, so this is making a gradient in our cell. So it makes an electrochemical gradient. So we'll have one side that's positive and one side that is negative. Okay, and this potential makes a voltage or a potential difference across the membrane. So we might have a bunch of hydrogen ions on one side, and so it makes it positive and negative on the other side. Or here we have all of our sodium and our potassium making a gradient, one side being different than the other. Okay, another type of active transport that is different is bulk transport. This is bulk dealing with movement of large quantities or large molecules. And so we can have endocytosis and exocytosis. Endo is when cells ingest something, it's going in. Exocytosis is going out. Okay, so endocytosis here, we have some molecules and it's being kind of ingested. This is the idea of kind of like our endosymbiosis, right, where we have a prokaryotic cell getting engulfed. That would be endocytosis. Exocytosis is the opposite, where we have a vesicle with waste usually, and then it is able to fuse with the cell membrane and release the contents. And both of these require energy because they are changing the shape of the cell, and that's going to require energy. Okay, so both of these are going to be dealing with our cell membrane, and we actually will lose or gain parts of the plasma membrane when it does this. You can see how in endocytosis, we have this part of the membrane then gets pinched off and becomes a vesicle. So think about, where do the membrane get more lipids from? What organelles are responsible for that? Okay, that's going to be our smooth ER. Remember, our smooth endoplasmic reticulum is what is synthesizing our lipids. They'll then go to the Golgi body and form vesicles that can then come up and just fuse with the plasma membrane and replace those phospholipids that have been used. So here we have our smooth ER, and we're making vesicles. So that's how we replace the cell membrane that's being used. Okay, our endocytosis can be broken down into more groups. So we can have phagocytosis and pinocytosis. And we can also have receptor-mediated endocytosis. So that's shown here in this image. So our phagocytosis, this is where the cell is ingesting large molecules, usually food. Okay, so it will engulf and then have the food in a vesicle. Our pinocytosis, instead of going out and around, the cell membrane will come inside and it will ingest material that way. Receptor-mediated endocytosis is similar to just our regular phagocytosis, but it's specific because it has receptors. When receptors are activated, then the endocytosis occurs. Okay, so this is showing a picture of a white blood cell engulfing a bacteria. So this is how our white blood cells kill bacteria. And here's a cute little thing about endocytosis and our phagocytosis. Okay. I have a quick little video that we will watch showing some of these, maybe. There it goes. Okay. So here we have two paramecium, and they're going to actually be ingested by an amoeba. So the amoeba here is so we're using phagocytosis to Okay, so our exocytosis, I have a video for that also. This again is forcing material out of the cell, exiting. Oops. So our exocytosis is happening here in this paramecium. They have to get it in focus. And you can see the contents of the cell leaving in exocytosis. This is how it's removing waste. Okay, so that's it for this class. There's some extra videos on Canvas if you need more help, and check out those focus questions. Bye, guys.